Hello, welcome to lecture number 2 in our series on physiological psychology. Dr. Paul Merritt. Today we're going to be talking about the action potential. The action potential is one of the most important properties to understand for physiological psychology. It's at the core of how everything we know and do works, is by the generation of this particular electric signal in the nervous systems. The action potential is a nerve impulse, so it's the electrical message that is transmitted down the axon of a neuron. It does not travel directly down the axon, but it is actually regenerated at points along the axon so that it is not weakened. So when an axon is generated, it has the same force and potential at the end of the axon as it does at the beginning. So there is no degradation as you would get, say, if you were pushing electricity down a wire. So the speed of a nervous impulse ranges from less than a meter per second to 100 meters per second, depending on the type of neuron and whether or not it has a myelin sheath. So a touch on the shoulder reaches our brain more quickly than a touch on our foot. Obviously, part of that has to do with distance. Part of that has to do with the type of neurons we're talking about. So the brain is not set up to register small differences in the time of arrival of touch messages. But in vision, movements must be detected as accurately as possible. So the properties of impulse control are well adapted to the exact needs for information transfer in the nervous system. So this nerve impulse control is particularly important for how signals are generated and how information is processed. So we don't detect subtle differences in touch messages, but we can detect very subtle differences in visual processing, because that's how we process movement, that's how we process changes in luminance, color, all sorts of other things. So this impulse control and this signaling nature is really important for turning on and off the messages from neurons. So this all gets accomplished by the properties of the cellular membrane of a neuron. So the first place we'll start is we'll talk about a neuron that's at what we call resting potential, when it's just sitting around doing nothing, waiting for something to happen. So messages in a neuron develop from disturbances of this resting potential. So at rest, the membrane maintains an electrical gradient we call polarization. So the membrane is designed to keep the electrical potential difference between the inside and the outside of the cell at about negative 70 microvolts. So a difference in the electrical charge inside and outside of the cell is this polarization. This is an important property because the way an electrical signal is generated is by depolarizing the cell, by opening up ion channels that allow ions to flow in and out of the neuron. And we're going to talk specifically about how the membrane maintains that process and how that process unfolds throughout the generation of an action potential. So the inside of the membrane is slightly negative with respect to the outside, approximately negative 70 millivolts. I think I might have said microvolts in the last one. It's millivolts. The resting potential of a neuron refers to the state of the neuron prior to the sending of a nervous impulse. So this is, again, prior to anything happening. This is just how the cell is maintained over time. So it just maintains that resting potential. So all of this gets accomplished, this resting potential gets accomplished by the properties of the cellular membrane that we talked about in Lecture 1. It has to do with the structure of the membrane itself, the lipid layer that has channels that allow the flowing of ions in and out of the cell itself. So on the membrane of the neuron, we have protein molecules, which are known as ion channels, which can open and close depending on circumstances, and we'll talk specifically about the kind of ion channels we're talking about. So the membrane itself is a phospholipid bilayer that keeps everything out except for through these protein molecule channels. We can record the amount of electrical potential within a neuron using what are called microelectrodes. We have an intracellular and extracellular reference microelectrode, and that tells us the electrical signal within that neuron. And it can also tell us when the neuron gets hyperpolarized or depolarized, etc. So the membrane is selectively permeable, allowing some chemicals to pass more freely than others. And this has to do with ions and ion channels. So sodium, potassium, calcium, and chloride are the four ions that pass through specific channels in the membrane. When the membrane is at rest, the sodium channels are closed. The potassium channels are mostly closed, but allows the sort of slow passage of potassium. Sodium and potassium are the two ions that are primarily involved in maintaining the resting potential of the membrane and in generating an action potential. Now, some drugs, such as benzodiazepines, influence the passage of chloride ions, and so can actually make a neuron less likely to generate an action potential through hyperpolarization. We'll talk more about that in a bit, but we're primarily talking about the sodium-potassium channels in the generation of an action potential. So these are the ion channels in the membrane of a neuron. They allow sodium and potassium to pass in various directions depending on what's happening with a cell at any given time. When an action potential is generated, those sodium channels open, allowing for sodium to flood into the inside of a neuron. We'll talk about that more here in just a moment. The other important part of the membrane is called the sodium-potassium pump, which is a protein complex that continually pumps 3 sodium ions out of the cells while drawing 2 potassium ions into the cell. So it's constantly exchanging 3 sodium ions, taking them out of the cell, and bringing in 2 potassium ions into the cell. This helps to maintain that electrical gradient, that electrical potential. It is one of the processes by which the electrical gradient or potential is maintained. These electrical and concentration gradients are the other part that help maintain the potential across the cellular membrane. The electrical gradient and the concentration gradient refer to the difference in distribution of ions that work to pull sodium ions into the cell. So the electrical and concentration gradients are trying to push ions into the cell, or sodium ions specifically into the cell. And so the reason those sodium channels are closed and the sodium-potassium pump is working is to keep the sodium ions out and potassium ions in. So that electrical gradient tends to pull potassium ions into the cells while at rest. So there are 2 processes that keep the level of potassium ions high within the cell at rest, and that is the sodium-potassium pump and the electrical gradient. The concentration gradient is trying to pull them out. So they slowly leak out carrying a positive charge with them because those potassium ion channels aren't completely closed. So that's why there's that potassium sodium-potassium pump working, and again the concentration gradient, because there are more potassium ions inside the cell than out, the concentration gradient pulls them outwards. So these are the gradients that are pulling things in and out of the cell. So potassium leaves the cell because of a concentration gradient. It enters the cell because of the electrical gradient, because they're positively charged and the inside of the cell is negatively charged. Sodium ions would enter if their channels were open, but those are closed at rest, so there are much more sodium ions outside than inside because that sodium-potassium pump is pumping 3 sodium out and bringing 2 potassium in. So that gets us to what happens in the generation of an action potential. The resting potential remains stable until a neuron is stimulated, which produces 3 distinct possibilities. We'll talk first about hyperpolarization. Hyperpolarization is increasing the polarization or the difference between the electrical charge of the 2 places, so the inside and the outside of the cell. A hyperpolarization is actually inhibitory. That is, it makes it less likely that an action potential will be generated. So anything that increases the charge away from 0, that is, so we're normally at -70, and so anything that pushes it further away from 0 is hyperpolarization, and it makes it less likely that an action potential will be generated, because what we really need is a depolarization for that to happen. So depolarization is the decreasing the polarization towards 0, and that actually is what happens when an action potential is generated, is depolarization. So anytime we hyperpolarize a cell, we make an action potential less likely. Anytime we depolarize a cell, we make an action potential more likely. It's complicated, I know. Now, why is this important? Because some drugs like sedatives, benzodiazepine sedatives, Xanax, Valium, Ativan, cause cell hyperpolarization, which is why they are sedating, because they make action potentials less likely. That's also why they alleviate anxiety, because of that hyperpolarization. So the resting potential remains stable until these things happen. The threshold of excitation is the level above which any stimulation will produce a massive depolarization. So the cell has to be depolarized past the threshold of excitation. And that will then cause a massive depolarization, which is an action potential. So depolarization makes an action potential more likely. Hyperpolarization makes an action potential less likely, because hyperpolarization is moving us away from the threshold of excitation, whereas depolarization is moving us towards the threshold of excitation, towards generating that action potential. So the action potential is a rapid depolarization of the neuron. The action potential threshold will vary from one neuron to the other, but is consistent for each neuron. Stimulation of the neuron past the threshold of excitation will trigger an action potential, and that is the generation of this electrical signal, this massive depolarization, which is how our nervous system functions. So what happens when a cell is depolarized is, if it is depolarized sufficiently, the voltage-gated ion channels will open. These are membrane channels whose permeability depends upon the voltage difference across the membrane. And so as long as it's -70 or higher, that is, it's much more hyperpolarized, these voltage-activated channels will not open. These are both sodium and potassium channels. When the sodium channels are opened, positively charged sodium ions rush in, and a subsequent nerve impulse occurs. Now again, if enough sodium channels are opened, we get enough depolarization of the cell that we get the triggering of an action potential, because we will have passed the threshold of excitation. So the critical thing to understand about the action potential is the movement of sodium and potassium ions. After an action potential occurs, the sodium channels are quickly closed. So the sodium channels open, sodium rushes into the cell is depolarized, because now we've got all of those sodium ions that were outside of the cell, creating that electrical difference, now inside the cell, and so we have depolarized the cell. The neuron is then returned to its resting state by closing those sodium channels and by the opening of potassium channels. Potassium ions flow out due to the concentration gradient and take with them their positive charge, again making the inside of the cell more negative than the outside. And then the sodium-potassium pump later restores the original distribution of ions. So it takes a little bit of time for all of this to occur. So the sequence of events at the action potential are: the cell starts to depolarize. Once it reaches the level of excitation, the sodium channels open. Sodium ions flood inside the cell. Quickly thereafter, the sodium channels close. The potassium starts to flow out of the cell when the potassium channels are then opened, and then the sodium-potassium pump starts to pump the sodium ions back out of the cell. So this is what this looks like. So we have the generation of an action potential, which massively depolarizes the cell. In fact, it goes above 0 for a while, so it becomes positively polarized. Then we start to get the return towards the resting membrane potential. So here's what's happening during this millisecond time. Sodium ions are flooding into the cell. So they flood into the cell, and then that starts to trickle off as the potassium ion channels open, and the potassium ions start to flow back into the neuron. And you can see over time that we get the return to our -70 millivolts. You can see that for a little bit of time the cell is hyperpolarized after an action potential, because we haven't sort of reestablished that membrane potential. During this time, this is what we call the relative refractory period, where it's more difficult to generate an action potential because the cell is temporarily hyperpolarized. So one of the things that we have to do is restore the sodium-potassium pump to get back to our -70 millivolts. So it does take time. An unusually rapid series of action potentials can actually lead to a buildup of sodium within the axon, because the sodium-potassium pump has not had enough time to get the sodium back out of the axon itself. And this can be toxic to a cell. And this only occurs in rare instances such as a stroke and after the use of certain drugs. So for example, ecstasy or methylene dioxymethamphetamine has been known to cause the kind of burnout of some serotonergic neurons, because it generates very rapid sequences of action potentials. One of the things that is useful from a pharmacological perspective is the blocking of sodium channels. This is actually how local anesthetic drugs work. They block sodium channels and therefore prevent action potentials from occurring. So this is how Novocaine and Xylocaine, those drugs your dentists use to numb your mouth, they're also used as topical anesthetics in other instances. So that's what that essentially does is it blocks those sodium channels so sodium can't enter the cell and an action potential cannot be generated. One of the things to keep in mind about the action potential is they function on an all or none law. That is, action potentials will fire or not. That is, once a cell has reached its level of excitation, it will fire. There are no big action potentials or small action potentials. One of the things that will happen is action potentials can actually back-propagate into the cell body in dendrites. So the dendrites then become more susceptible to structural changes. This is part of our long-term learning, is through the structural changes of the dendrites. The all or none law states that the amplitude and velocity of an action potential are independent of the stimulus that initiated it. And this is important because action potentials are equal in intensity and speed within a given neuron. What that means is we cannot code for how bright a light is or how loud a sound is or how salty our food is based on the intensity or velocity of a single action potential. These require differences in firing rates of neurons or differences in the activation of several different kinds of neurons. So a single neuron itself can't code for much because all it can do is fire or not. Sort of like Morse code. You don't have differences in tones. You just simply have the same intensity and amplitude within any given action potential. So after an action potential has fired, a neuron has a refractory period, during which time the neuron resists the production of another action potential. There are two refractory periods, the absolute refractory period and the relative refractory period. The absolute refractory period is the first part of the period in which the membrane cannot produce an action potential. That's because it just simply cannot, it's already depolarized, so it cannot fire an action potential during that first millisecond. The upshot of that is that the fastest a neuron can fire is about 1,000 times a second. So anything that might require a neuron firing faster than that has to require multiple neurons to code for that kind of stimulus. The relative refractory period is the second part in which it takes a stronger than usual stimulus to trigger an action potential, because at this point the cell is hyperpolarized and requires a much stronger stimulus for the next 4 or 5 milliseconds. To sum up what's going on during all of these periods, so we have a stimulus that causes the firing of an action potential. Sodium enters the cell, and we start to get repolarization with the opening of the potassium channels. Okay. We then get a brief hyperpolarization. It's sort of an undershoot. We've overpolarized the membrane, so this is our relative refractory period, whereas this is our absolute refractory period, and then we get back to resting membrane potential, and everything is as it was before. The propagation of an action potential occurs differently depending on whether or not it's a myelinated neuron or not. In a motor neuron, the action potential begins at the axon hillock, swelling where the axon exits the soma. Propagation of the action potential is the transmission of the action potential down the full length of the axon. The action potential does not directly travel down the axon. However, as the action potential propagates down the axon, ion channels open and close. So it's being propagated down kind of like a wave. So the way this happens is ion channels open and close down the length of the neuron, which is why there is no loss of electrical signal, because the electrical signal is generated at every step of the way. So this is how this works. We have a stimulus, causes an action potential. We get the sodium and potassium channels opening and closing, and this propagates its way down the axon as an action potential. The myelin sheath is an important part of this process. The myelin sheath of axons are interrupted by short, unmyelinated sections called the nodes of Ranvier. These nodes of Ranvier are what allow action potentials to propagate down myelinated neurons so quickly. That myelin is an insulating material composed of fats and proteins. At each node of Ranvier, the action potential is regenerated by a chain of positively charged ions pushed along by the previous segment. So those voltage-gated ion channels open at each node of NVA. It looks something like this. So we have the myelin sheath covering all of the ion channels. So only at the nodes of NVA is this action potential generated, and so it's generated at each node. We call this saltatory conduction, which is the jumping of the action potential from node to node, provides rapid conduction of impulses, and conserves energy for the cell so that the cell doesn't have to propagate the action potential down the full length of the neuron. Rather, only has to go down the small places in between the myelin sheath. Multiple sclerosis is a disease which causes the demyelination of the motor and sensory neurons. So what happens is, for some reason we don't yet understand, the body's immune system starts to eat away at the myelin sheath. And as a result, we get loss of sensory and motor functioning because we get slowing down of neural impulses because they don't travel as quickly without that myelin sheath. So this is associated with poor coordination and sometimes visual impairments. So here you can again see the local current flow underneath the myelin sheath, and then of course the opening of ion channels at the nodes of NVA. Final note in this quick introduction to the action potential: there are local neurons which have very short axons which exchange information with only close neighbors and do not actually produce action potentials. When stimulated, they produce graded potentials that vary in magnitude and do not follow the all or none law. And they will either depolarize or hyperpolarize in proportion to the stimulation. So these are interneurons contained in local places, are not primary motor or sensory neurons. And so they follow a little bit different process. So our next lecture will get into the process of synaptic communication and how this action potential is generated by the transmission of neurotransmitters across the synapse.